Hi, welcome to our video on CRISPR. CRISPR stands for Clustered Regularly Interspaced Short Palindromic Repeats, and it is a method used to genetically modify DNA. It originates from bacteria, specifically E. coli, as a defense mechanism against bacteriophages. CRISPR is a catalog of viral DNA stored in the bacterial genome that allows the bacteria to create special enzymes that destroy specific pieces of viral DNA that the bacteriophage has injected. When the bacteria encounters viral DNA it hasn't seen before, the CRISPR system is able to insert that viral DNA into the existing bacterial genome, storing it as a sort of memory bank so that it can be used later. The reason that CRISPR is useful is that scientists have been able to hijack this bacterial process to be used however we want. The Cas9 protein is the main part of the CRISPR system, and it splits apart the DNA as shown. It cuts both strands of the DNA wherever the DNA matches the CRISPR RNA. And by adding our own CRISPR RNA, we can cut the DNA wherever we want. The cell fills in these holes with mutations, which break the gene and deactivate it. Alternatively, the CRISPR system can also be used to insert a new strand of DNA called host RNA that is added to the Cas9 protein, which adds this replacement RNA in the place of the cut and is filled in to make inserted DNA. This is important because CRISPR allows us to fix both malevolent DNA and improve existing DNA. For example, it can be used to provide resistance to AIDS. Normally, the HIV/AIDS virus is able to bind to a receptor on the outside of the cell and inject its virus. But with CRISPR, we can change the DNA that codes for the receptor, changing its shape and preventing the AIDS from entering, making the person resistant. A second thing we can do with CRISPR is enhance existing structures, like fruit, for example. We can improve the fruit to be larger and more nutritious by editing its DNA with CRISPR. Thank you for listening, and we hope that you enjoyed our video on CRISPR.